Welcome everyone. Welcome everyone to our the Center for Health AI seminar today. So we're very excited to have Dr. Mangul to give a talk here on computational data-driven immunology. So as you know that Dr. Mangul is an assistant professor in the University of Southern California School of Medicine, and his lab has been developing very cool and novel computational methods to link the genomics and the immunology and for translational research. So without further, welcome, and the floor is yours.
Okay, thanks a lot. Let me share my screen. Okay. Can everybody see my screen?
Yep.
Great. So yeah, first of all, really excited to visit. I guess that's a new reality, online. I think, I was hoping to get some snow or see some snow. We don't have that in California, but I guess, probably next time. So yeah, what I want to talk about, I think I would like to talk about kind of opportunities and challenges in computational driven immunology, at least the way I see it. So I recognize myself as the newcomer to this field. So how I'm looking at the field, I think I'm looking from the computational perspective. And I think it's contrasting that what was happening in genomics and what I believe yet to happen in immunology. So I would like just to give my view and kind of get your opinions on that. I think I want to start and recognizing the value of the data in that it's taken the central stage across many domains, and that happened in genomics and other domains, but that I believe yet to happen in immunology, and there are various reasons for that. But despite that, I think the data is essential, and the availability of the data and richness of the data enables many questions to be answered. And on other hand, it enables kind of opportunities to people like myself from the computational background to contribute to the field and perhaps enable novel biomedical discoveries, being that in immunology or other domain, through better bioinformatics and through better analysis of the data. And I think one effect of that is already happening in immunology. I think the databases which currently exist, for example, for immunogenomics, are being challenged from computational perspective in terms of are they complete, are they serving diverse population equally in that sense, and what can we do by let's say bringing the high throughput technologies and bioinformatics to this field. And on the journey, I think the wide community of computational researchers are yet to come for this domain. So I remember myself when I joined PhD long ago in 2008, when I came from pure applied mathematics background, knew nothing about genomics, I mean, except probably high school knowledge. But I remember my PI back there introducing me RNA-Seq. So basically what he did, he drew a long line, being a genome, and a couple of boxes on that line being exons, and short lines, and said, okay, those are the reads. How about we inference the gene expressions, right? So I didn't understand much, but I understood the logic. I understood the strings. So I think in immunology that will be perhaps more complex, you cannot simplify at that level. But I think in the genomics, that's one reason so many bright and talented researchers switched the fields and came to genomics and brought the expertise, because it was easy to jump in. Like RNA-Seq, there is 100 or more methods just doing quantification. So it's quite diverse and intense field. I think that yet to happen in immunology, and I think there are many reasons why it didn't happen. I guess perhaps it's more complex, more knowledge is required, but technology yet to scale, yet to come to the field. So I think overall we all recognize the flood of omics data being around, variety of technology being developed. Some of them are great, some of them yet to be kind of polished and so on, but we have a lot of data, right, generated by those technologies, and the data is not necessarily perfect, as all of I don't need to kind of tell to this audience, so that's what I see bioinformatics is the frontier between noisy and large data sets on the left, and then meaningful, biologically relevant and clinically relevant interpretation of such data sets on the right. So I think that's the last slide to show, I think giving the availability and accessibility of the data, I think that is shaping the field, that we have enormous number of data being generated, and that data need to be processed, and the availability of the data, in major part enables population level surveys, like in genomics. I think population level genomics is around. However, population level immunogenomics is not around due to lack of the data. So that's kind of yet to happen. But I guess talking about technology, I think doing that, like having the availability of technology, we have generated as the global community quite a lot of data sets, around like 8 million or so samples being available as we speak. Some of them are actually available, freely. Some of them kind of semi-available, so you should write some note and say what you want to do with the data. And some of them, I guess, intentionally designed not to be available. So one example, I guess, is data from or other countries where I think you need physically to come there and be a resident, and then they will give you the key to the secret room where you can analyze the data, right? So there is quite a variability in terms of availability, but majority, or at least substantial part of the data is available, so we can reuse that. So talking about computational data-driven immunology and in general, kind of computational and data-driven research, I think there's a new domain where availability of the data sets and technology enables new types of research being happening, which I refer as the dry lab research, where like what we do in my lab, we don't run a wet lab component. So instead we rely on the public data. So we do reanalyze the public data, or we rely on the collaborations with the immunologist or other wet lab faculty to collaborate on the data sets. And you might ask, why do you want to reuse the secondary analysis? I think many people in this audience are doing that extensively, perhaps other people less so, but I think there is a tremendous value in reanalyzing the data. And for various reasons, like you can do just meta-analysis, basically increase your sample size and hope you will get the signal which was originally not accessible to the people for the original study with the limited sample size. I guess capturing real world heterogeneity in the data, that's extremely important. I guess being in Los Angeles here, it's less of the issue because we have diverse populations coming to the hospital. However, in the areas which are less heterogeneous and more homogeneous in terms of diverse populations, I think that might be the issue, because it might be the issue that recruiting the patient is only possible from the certain ancestry group. So I think combining the public data perhaps with the local data might increase heterogeneity. And not talking about repurposing, which I will talk today about, repurposing the technologies. Technologies were designed to answer one question, but I doubt, or at least I disagree, that data is, like one data set, one question. I guess the data set can trigger multiple questions to be answered. So I think in this way, like the best way to think about the secondary analysis is existing data and novel biomedical discovery. So on that note, just to bring everybody on the same page, so what we mean by the primary and secondary analysis. Primary analysis meaning that it's traditional wet lab research where you recruit the patients, you get the samples from those, and you run any kind of throughput technology like being sequencing, and you would do the analysis, find your results and biomarkers and things like that, and you would publish. Along that process, luckily, I guess, for computational people, I'm really glad that we agreed to share the public data, right? So many journals require the data to be shared as you publish your results, right? So what that means, we have accumulated, as I mentioned previously, quite a lot of data sets which are publicly available. So what that means, that kind of right of those data sets being available, that means we can do secondary analysis, right? So we can skip kind of the part with recruiting the patients and sequencing and other parts, and directly acquire and download data from the public repositories and get the data and combine that and repurpose that and do the analysis. And perhaps discover something new despite the existing data. So I guess by doing that, there is a lot of opportunities and challenges as you do, computational research, data-driven research in biology and immunology. So we have recently, we're curious about that. So what we did, we have interviewed, I would say, variety of the scientists which are pursuing dry lab career on the high level. And we asked them, what are the challenges and opportunities? So we are planning to integrate that in any sort of opinion paper or commentary, just because we were so curious and we realized there is little, there are domains where people agree, but there is quite many domains of computational research where people disagree, and the value of the data and the value of secondary analysis, and is it like a feasible approach to do. So I hope in a few months, We have finished the interview phase. So we have interviewed each of those scientists and many others. And we will summarize that anyhow. We are still thinking about the format. So as we go, like I think one inspiration we have got in terms of secondary analysis, I think was Casey, which is here in terms of like, parasite award. I think it's like my favorite example how you bounce back, right? Like how the paper that New England Journal thing came out, and then you make a nice analogy and you make a word and things like that. So I'm pretty sure everybody knows, just for those, just to make sure, it's like the Parasite Award recognize the people who do good job reusing the data. So, and then that's outstanding. So, and then for us, we were curious, but what is the scale of that? Like we know the winners, and thanks for Casey, like recognizing them and the committee to selecting the winners of secondary analysis, but do we know the scale? How many people in the world do secondary analysis? And that's something we have been actually collaborating with Casey. So we have got, Casey, maybe we didn't share the results with you, so I would share some preliminary results now. So basically what we did, we have looked at all the PubMed publications and we have extracted the SRA and GeoIDs. And then, luckily for us, they're unique. However, we didn't know what is the paper doing primary analysis versus secondary analysis. And then it's almost impossible to know. There is quite a lack of the structure in the publication, or the structure is quite different across journals to kind of conclude that. So what we did, we assumed that our database is complete, meaning that the very first paper mentioning the datasets will be the one introducing that. Other words, doing primary analysis, and all the rest of the papers will be secondary analysis. So doing that, we actually compare the publications doing SRA, uploading the data to SRA and reusing that versus Geo. And what you see, I guess for SRA, there is a good fraction of the papers doing secondary analysis, like dry lab research. But for Geo, it was remarkable. There are actually slightly more papers doing dry lab research, like secondary analysis comparable to primary analysis. If you're curious about that, how did that go across the years? So those are the plots. So each plot is not cumulative, so it's per year bar plot. And then, so what we can see for SRA is more or less stable across the years. However, for Geo, the trend was reversed. So in 2016, it was already slightly more papers doing secondary analysis than the primary. I guess the good news, or like, I don't know, is it good or bad news, but majority of the data set are yet to be explored for secondary analysis. So 60% of SRA data sets not being touched, meaning they never participated kind of in the secondary analysis. Oh, sorry, for Geo, it was 60%, but even more for SRA. So there are some superstar data sets and some data sets which are quite reused many times. And some of them come from, for example, for Geo, they are coming from some cancer cell line. I think in particular that was the breast cancer. For SRA is less clear because you have a variety of the human data, animal, bacteria, and the plants. So somehow the most reused data set was cotton. Not sure for what reason. Maybe there is a single cotton data set, nothing else. So people reusing that one, but that yet to be explored. We are curious in general, what is driving the reusability? What are the factors which make some data sets superstar and others not? So perhaps the technology, like perhaps nobody's reusing very short reads like 36 or something like that, or solid, right? You would probably don't want to reuse solid color base. But also sample size, like I think we do observe that nobody's reusing the data sets which are smaller than 10 samples. So that's very preliminary. We kind of actively working on this, so we hope to wrap it up in the paper soon. So on that part, just I wanted to set up the ground in terms of like data-driven research in general. So as you can see, there is already interesting observation, like reusability of the data varies across different repositories. Of course, we don't know exactly the answer, but I think we know that. Basically, Geo, you can get access to the gene expression, mostly, right? So meaning that you can reuse the data quite easily. You can say which samples do you want, which genes do you want, and you will get the metrics. And you almost can import that metric in Excel. So that's very appealing for the people with the less computational background and less kind of computational capabilities of like access to the clusters and so on. So it's exactly opposite for SRA, where, like to reuse that data, and I already spoke to a few people from here during the interviews today. So it's a pain, right? So you need to have expertise to remap the reads, and perhaps you need expertise or like access to the cluster and things like that. And actually, if you think about that, that's a lot of like redundant work being done across the globe. If you think about that, you want to study a particular HLA loci, and all you care about the mutation in that. But the data is actually raw FASTQ file. So you would need to remap the entire, like the entire sample, and then perhaps extract just the reads from that loci. So of course it's not great. I mean, it's actually very laborious, time consuming, and perhaps impossible to process 70,000 sample RNA-Seq samples being publicly available. So I think that's one of the reasons. If you know any others, let me know, but we are thinking about that. Okay, so I think just over my postdoc work at UCLA and now the faculty for 2.5 years at UC, I have focused my research across a variety of domains, presented kind of here. So today I want to talk about the particular domain, about computational immunology, and present a couple of the methods we have developed and kind of say where we are planning to go and where we think computational research can make a difference in immunology. Okay, just to bring everybody on the same page, that we will be talking about the adaptive immune system, which is encoded by several loci distributed across your genome and several chromosomes. We talk about Ig, immunoglobulin, couple various chains like heavy chain, copper chain, lambda chains. We also talk about TCR, T-cell receptor. So I think when I first kind of realized it's an outstanding machinery of immune system, which is outstanding from kind of computational perspective, like it's really was fascinating to realize how things are working. So basically we'll be talking about the collection of the V genes, variable gene, D genes, diversity genes, and joining genes, J. But we have a limited number of those on your genome. So if you try to recombine them, you will get not that high immune diversity, meaning you will not be able to produce any sequence you would like, but that's essential for the immune system. So what else is there? Actually, you're also allowed to insert non-template insertions at the junction point between V and D in D and J. So that machinery, that's one reason why T-cell and B-cell repertoires are so diverse, 10 into power 15 distinct sequences, right? So, and literally being able to generate any sequence. So a couple of definitions. So we'll be talking about complementary determining region 1 or CDR1, CDR2, and CDR3. In particular, we'll be focusing on CDR3, complementary determining region 3, and that will be our proxy to define the clonotype. And the collection of the clonotypes of the particular individuals will be referring as the repertoires. So what that means in terms of the reads. So if you think about the reads, which are non-targeted reads coming from the whole genome sequencing or coming from, let's say, RNA-Seq. So the reads which are in black, they are mapped, right? If you run star for let's say, for RNA-Seq, or you run BWA for let's say, whole genome data, since the V, D, and J genes are part of your genomes, they will be mapped. However, the recombinations of those genes, they are not part of your genome. So the reads which are in gray will be unmapped. And this is exactly where we will be looking at. We'll be looking at those genes, try to see if we can assemble some clonotypes, and having those clonotypes, can we quantify the repertoire in terms of clonality or diversity? So you might ask, why not use the proper technology? The proper technology will be referred as AIR-Seq, or sometimes referred TCR-Seq or BCR-Seq. It requires a specialized protocol, which is available. However, the number of samples of those specialized protocols is limited. My guess, or my estimation, is several thousand samples being available versus 70,000 samples available to RNA-Seq. So those technologies did not access the scale. So if we want to think about immunome, and that word perhaps is less familiar to the audience comparable to the genome. So the reason we are not talking about immunome, because we are not there yet, and actually it's substantially harder to reconstruct the immunome, right? The reason we know the genome is because we are so similar to each other, right? So we are different quite in a few places in the human genome. So that's why it was kind of enough to reconstruct to do good job once, basically, to take an individual or several individuals to really do good job and get the reference genome. And that's kind of enough, and we keep using that. I guess there is a discussion, should we use it across population? Should we reconstruct population-specific reference genomes? But I would say that will not have a major effect, right? You might increase your bioinformatics results slightly, but perhaps not dramatically. However, that's exactly opposite for human immunome. We are more different from each other in terms of adaptive immune system comparable, how similar we are, right? Meaning that to really reconstruct the human immunome, we should probe every single individual or majority, let's say, of individual on the human, on the earth, meaning so far that's not feasible. So what can we do? We assume that we said that, okay, let's use the most kind of widely used technology nowadays and try to repurpose that technology to reconstruct the immunome. And that technology is RNA-Seq and whole genome sequencing. So we would be talking about, is that technology feasible to extract the clonotypes and make a first step, a small step perhaps, but the step towards the human immunome. So if you think about RNA-Seq, you know, like as you got the reads, or the fragments which you sequence, you know, like you shred your DNA, get the fragments, sequence those. I think the traditional bioinformatics approach will be take those reads, map it to the genome, get your gene quantification, and get the gene expression, and answer interesting biological questions like differential expression genes, like RNA splicing. If you're a sophisticated bioinformatician, you can try to assemble novel transcripts. So what is in common between all of those approaches? That you're using only the fraction of the reads, and another fraction of the reads go literally to waste. Right, so that was one topic I was interested in. Can we explain all of those reads which are never used? And actually we did. We did explain all the reads except 0.1%. And actually we share those reads across several samples. If you're interested to improve our work, please do. But what I want to talk today about, I want to talk about that particularly reads which are coming from recombined B and T cell receptors. And then it's a small fraction of those reads, but we would argue they're meaningful, they're important, and they can be used to quantify the immune system. Now, why they're there, right? So the reason they're there, because as you sequence the sample, you do have B and T cell present, typically present in the samples like whole blood or PBMCs, perhaps even in the larger fractions. So they're there, and they have their own genomes with the recombinations, right? With the particularly recombined VGJ genes. So we will, as we sequence, so besides kind of normal human reads coming from the genome, or genomics reads, perhaps better say, we will also get the reads coming from those regions, I mean, from those genomes of B cells, so they can be used to quantify the adaptive immune system. Okay, so I would like to kind of highlight, give a flavor, at least one part of the tool we have developed. So the tool is called ImReP, Immune Repertoire Profiling. It's quite a simple tool, but remarkably, it was doing quite great. And I think due to few observations we have done. So the paper is in Nature Communication, published, I guess, last year or 2 years ago already. Okay, so what we do, I will just walk you through, spend 5 minutes here, try to kind of give you the flavor of the algorithm. So as we have the reads which are unmapped, right? Because we don't need to examine actually the reads which are mapped. If the reads map to the human genome, we say they're genomics, no need to examine that. However, the reads which are unmapped, meaning they couldn't be mapped to the human genome, they perhaps contain some reads which are coming from recombined B and T cell receptors. So what we do, we convert the read into amino acids, to all the frames. And what we do, we actually use the definition of the CDR-free sequence right away. So if you're talking about all the chains except heavy chains, so that definition will say that the CDR-free sequence is the sequence which starts with cysteine and ends with the phenylalanine. Okay, so we know that. Can we use that knowledge prior? Because other methods actually were using that in the very end. They were trying hard to make sophisticated bioinformatics algorithm, perhaps assembly and other things, but then in the end, only taking those sequences which start with cysteine and phenylalanine. And we said, why not use that in the very, very beginning? And this is what we did. So we look at the reads from the left to the right until we find cysteine, and the same from the right to the left until we find the phenylalanine. Okay, so that will be our candidate CDR-free sequence. So white candidate, actually we know nothing about the flanking regions of the read, right? So we would need to check those. So we'll take the flanking region, beginning of the read until the beginning of the CDR-free sequence, And we will check to all the V genes we know one by one. And things, they are actually beautiful because based on our observations in the database, the 3 before last codon is always cysteine in the V gene. So you just need to kind of put cysteine above cysteines. You don't even need alignment. So we check every V gene one by one until we find the match. So in this case, V3 was a match. And we do exactly the same things for the J, right? So J4 will be the match. So this is no longer the candidate CDR3 sequence, but rather the correct CDR3 sequence. We also do several steps, like if the read is short, so it doesn't overlap both V and J, we still use that. So we have some steps for that, as well as correcting the errors. But that's kind of the core of the algorithm. Okay, so what we did first, as usually, I guess, as all the bioinformaticians do, we use simulation data, kind of like with variety of parameters like read length and the sequence and the coverage to kind of prove to ourselves that we are doing good. And indeed, like I think in the majority of the cases, we were overperforming the existing methods. So if you want to summarize both recall and precision into one metric, like F-score, the results are even more evident. So we are in blue, our method. All right, so as we did simulation data, to me, So to me, like doing simulations data is never enough, like that's just the start. So I mean, in ideal case, we would like always get access to the real gold standard data sets. So and we found one. So we found 18 tumor biopsies being sequenced by both BCR-Seq and RNA-Seq. So we have treated BCR-Seq like a proper protocol to study the B-cell receptors as the ground truth, and RNA-Seq, which is here on the x-axis, as something we want to examine. So here, every dot is the clonotype, and then the value on the x-axis, that's a relative abundance being estimated by us, by our method, based on the RNA-Seq data, and the y-axis, the relative abundance, which is truth, right, estimated by BCR-Seq. So what we can see, we're doing quite well, right, with a few exceptions, but overall, the results were quite good. We also asked the question, what is the fraction of the repertoire? In other words, the collection of all the clonotypes we are able to capture. So, and then we compared to another method, which is in orange, the state-of-the-art method, mixer. So what you can see that we are capturing, first of all, more than existing methods, meaning that we can capture more clonotypes, but also interesting, there is some interesting trends where as, like we are capturing mostly highly abundant clonotypes, and as the frequency of the clonotype decreases, we have harder time to capture that. So overall, we are capturing above 50% of all the clonotypes. But if all the clonotypes would be above 0.06%, right, we'll capture all of them. Now, why it's happening? I think there are two reasons which we are currently exploring. So one reason could be might be the failure of bioinformatics tools, or you just don't have enough reads, right? You don't have enough reads to conclude that there is a clonotype, and you don't want to risk, you don't want to introduce false positive. Another reason is that it might be just no reads coming from the rare clonotype. So there is no chance for bioinformatics method to assemble that. So actually we are trying to estimate now, as we actually taking the next step and running kind of a proper benchmarking on this, we're trying to estimate kind of the upper bound of the bioinformatics tool. So what we do, we take the truth, which is the true clonotypes, and we do map the RNA-Seq reads on those clonotypes. And we look to see which ones of them at least have one read being supporting that clonotype. That will be upper bound bioinformatics. You cannot do better than that, right? If you don't have the reads, you cannot make a difference. If you have one read, you can try. But that will be the upper bound of bioinformatics tools. And we are curious to know how far away we are from this upper bound to kind of leverage our effort. Should we try harder and improve our tool? Or maybe we reached the highest point and there is no chance to improve. Okay, so as we get the method and we got kind of rigorously validated, at least to the extent we could with existing data, we were looking to see where can we apply that and get some interesting insight into immune system and adaptive immune system. So what we did, we get, as the part of the GTEx consortium, so we get access to GTEx data. For those of you who are less familiar with GTEx actually so it was concluded, I guess, a year ago. So it was a huge effort to kind of probe diverse human tissues post-mortem. So it was like 53 or so body sites across more than 1,000 individuals. And the primarily goals was to look at the gene expression patterns across the tissues, across individuals, look at the EQTLs, cell type specific EQTLs, and things like that. But we took a different approach. We said, okay, can we use that RNA-Seq data to look at the immunoglobulins and see the distribution of those across the tissue types? And this is what we did. We have applied our method across more than 1,000. Usually it was, I think, it's 1,000 samples, however, no, 1,000 individuals, but that comes to 10,000 or so samples. So we did that apply. So in general, we have got 3.6 million distinct CDR3 sequences. And that's a distribution of the number, what we call diversity of the repertoire. That's just the number of the sequences being per 1 million reads being assembled. So what you can see that the proper secondary lymphoid organs, I mean, that's obviously like the spleen and small intestine, terminal ileum, things. I mean, you have a lot of diversity there. However, less trivial was to see quite increased diversity in minor salivary gland and the colon and things like that. So what we see that many of the body sites which harbor some clonotypes, they actually have some mucosal sites. We have used brain and the nerve as the negative controls, expecting to see nothing there, and indeed we did see nothing in terms of the clonotypes. I think we see a couple of reads in the nerve, I mean, couple of clonotypes in the nerve, but I think that's due to the blood contamination, right? It's very hard to kind of purely extract the tissue and the blood is there, so that might be causing kind of contamination. So as we move forward, I guess like discovering some biological insights, we also thought, can we deliver to the community in the sense of the atlas? And we did. And I was surprised to, when I joined this project many years ago, so I was surprised to realize that there is not a proper database where you can have a knowledge about, is this clonotype public or private? Meaning, are you observing that clonotype for the very first time? Or was it observed before? And if so, was it associated with any diseases? So this is what we are on the path to. We would like to have a database, and this is just a preliminary kind of step where we record the information about all the clonotypes we know about. So far we have a simple kind of information about is this clonotype, which tissue it was observed, how many individuals it was observed, like one, which is private, or was it observed across many individuals being public. We also have access to the pathology notes, which are obviously useful. So we have looked at thyroid tissue in particularly, and we have asked the question about Hashimoto disease. And we have compared the number of clonotypes, IgH, immunoglobulin heavy clonotypes, across the kind of stages of the disease. So as the severity of the Hashimoto disease was increasing, we did observe more and more clonotypes being present in the sample. We have also looked at the sex differences across the tissues, and in majority of the cases, we observed none, except I guess the tissue you would expect to observe in the breast. And then as you would expect, the females will have more immunoglobulin heavy clonotypes comparable to the male. And then actually we were happy to find one outlier. In the male, the person was diagnosed with Ganyma-Pastia disease, where I would expect that you would need, you would expect to have more clonotype because the function or the shape of the breast of males becomes like females, so similar to that. So moving forward, we really look to scale. So we'd like to apply our approach across all the public data. And I think it's 70,000 or maybe even more, 75 or so thousand samples being available as we speak. So we'd like to leverage those rich resources and make a first step, what I've mentioned about Immunome, looking at the clonotypes across distinct populations. I think this kind of in the flavor of what 1000 Genome did at the time, observing the alleles which are different across the continent, across the ethnical and ancestral group. I think we'd like to do the same, but now leveraging the public data. And we have some preliminary results. We are collaborating with Privesh Harti from Stanford. So we have run some data sets, and we already observed some interesting results. For example, V gene usage is different for diabetes type 2. There's some interesting signature for the T cell receptor alpha chain. But that's just the start. And I think, interestingly, many samples from the public databases, they have good annotations. Many don't, but many do have. So we know the diseases. So that's a quite rich resource for that. Okay, so talking about the immunome and kind of diverse population, we are kind of making our first steps into that. And we have started by kind of setting the ground, and we realized there is limited discussion about what is the availability of the immunogenomics data across populations. We all know in genomics, right? Perhaps you saw the papers about that, that the genomic studies are really Eurocentric. The majority of the sample, like 80% or so, GWAS dataset are European origin. But it was no information like that in the immunogenomics. So we were curious to do that. So what we discovered that it's kind of very similar. That majority of the samples in at least TCR-Seq, this is what we have looked at. We have looked around 3,000 samples, and then this is what we see, that majority are Eurocentric, that is dominated by Europeans. And there is some interesting insights, meaning that if you look at the number of participants, so indeed Europeans are quite dominated, but if you look at the number of studies, less so. So the reason actually is that European-based studies, they tend to have more samples, right? So despite kind of a substantial number of, let's say, Asian-based studies, they just limited in the sample size. But we also discovered another problem, which we kind of anticipated. So I know some of you are working on the metadata and availability of metadata, or completeness and standardization metadata. So we have seen actually that the vast majority of the studies, they do not share information about ancestry, self-reported ancestry information. So what we did, we actually approached everybody and asked them to share the data, and we succeeded in 20% of the cases, right? So people were quite nice to share that. And actually we discovered there are multiple reasons why people don't do that. Let's say IRB, if you didn't put the correct IRB, where you say, I would like to share this, like perhaps you cannot share it, right? So we thought about, like, I think it kind of aligns with kind of our observations overall about availability of the metadata, which we recently published in Genome Biology, concluding that at least 40% of the metadata is lost, right? So it's not shared. So it's kind of aligns with that. And I think it's an interesting direction of research we are taking, like how to improve it, right? What can we do? Should we maybe run NLP to mine the text of the publication? Should we cultivate the beta culture? What will be the incentive for the people generating the data to do good job on the metadata? I think one success that we are doing good job, or at least relatively good job sharing the data, that the many journals were quite aggressive in that regards, mandating the data sharing, but there is no mandating of the metadata sharing. So that's to us an interesting question. I think one radical idea we have got, can we reverse it? Can we mandate certain phenotypes collected, and you should justify why you're not collecting those phenotypes instead of justifying why you should collect the phenotypes? But I think it's an open kind of area of research, and we are definitely interested to move that way. Okay, so one observation we have done, that it's not just lack of the data across the populations, but it's also the database, like immunogenomic database, like similar to what you think about the reference genome, but like in terms of V genes, it's actually disproportionately incomplete. It's definitely incomplete, but it's also disproportionately incomplete across the population. So, and the reason for that, the database was built quite a while ago in France, and I imagine primarily based on European populations. I think there is some component of Asian ones, but less so other populations. So first step we would like to take, we would like to assess that. So what we did quite a simple approach. We take samples where we know that the genetic-based ancestry by running whole genome data or genotype array. So we can split individuals into the groups. Now, we just match, and then we're gonna match, or we are matching actually the TCR-Seq reads with the reference and just counting number of mismatches. And the hypothesis is more mismatches you have, perhaps it's coming from the novel alleles, which are not present in the database, assuming the noise, the errors are equally distributed across Europeans and Asians in our case, which is okay assumptions because we did collaborate with, it's our data, so we generated in collaboration with Huda Alashka from UC. So we have a control over that. So we already see some interesting results. So if you look at the box plot, so every dot here is the read. So we just observe number of mismatches between the read and the reference. So on the right, on the left, this is mismatches, and on the right, those are deletions. So you can see in both cases, Asian individuals have more deletions and have more mismatches. So that's just the start. We only have 6 samples, but obviously the one we generated, but we look to scale and use public data as well as generate more data. So as we move forward, like kind of recognizing that the current databases are disproportionately incomplete. So we'd like to ask the question, how computational immunology can help? So, and we think it can. So we can apply bioinformatics method across the TCR-Seq data, and we can try to infer novel alleles. Of course, that needs to be benchmarked and validated, but I think it's a good kind of direction. There is a challenge. I guess the relative performance of the tools which are capable of inferring novel alleles are yet to be known. But I guess where is the challenge? There is opportunity. And I see that, at least personally, as the opportunity. I think there is opportunity, first of all, to benchmark those tools using comprehensive gold standard, the ground truth. And we have quite a bit of experience of running the proper or rigorous, let's say, benchmarking tools. And we have previously formulated the principles of how to do the proper benchmarking and what it takes. So doing that, I think the very first approach you can propose or you can think about, you say, okay, why don't you simulate the reads? Sure, we will. And this is like the setup we did, right? So we can simulate the reads from the full database and then kind of manually remove some genes from the database, kind of creating this incompleteness. And then we can ask the question, what should be original completeness of the data? So you can kind of complete it to the full extent. Let's say if your database is incomplete 50%, can you reconstruct another 50? Maybe not feasible. Maybe it needs to be complete at least 80%, right? So those are the questions we can ask them with simulation data. However, as you know, simulation data is always oversimplification of reality. So we think about the real data, right? So what will be the real gold standard benchmark? We need to recruit the patients. We need to perform AR-Seq or TCR-Seq or BCR-Seq analysis or sequencing and analysis, run the tools which will infer alleles, and perhaps take those alleles, right, and go back to the wet lab and run any sorts of PCR or Sanger and validate them. Ask the question, are they there really in the sample? Sounds like a great approach. However, there are limitations of that. How about false negatives, right? How about the alleles which are there, but were never kind of reconstructed? Will you know that? Perhaps not. So, but then if that's the case, I think you can do precision of your tools, but not having false negative will limit you from doing sensitivity. So how can we fix? So our idea is like that. So those are all your alleles, right? In orange are alleles covered with at least one read. But I would argue this is what we care about in terms of bioinformatics, right? So if the allele is not covered at least by one read, there is no hope to reconstruct that. So perhaps, we will never be able to reconstruct those alleles not covered by single reads. So then we can maybe reduce our gold standard or kind of hypothetical gold standard to the smaller one and say we will have a gold standard which at least of all alleles which are covered by at least one read. And that's possible to do bioinformatically. You can just map the reads onto the known alleles and see if they are covered. So that, we hope, that setup, we actually applied that recently for structural variant detection and it was quite successful. So we'd like to translate this in this domain and we hope it will allow us to do false negative. Okay, so kind of like moving towards the conclusion. So I think the barriers in data-driven immunology, I think they're similar as in genomics. Maybe there is some specifics. I would say barriers are like lack of computational capacity. And then of course it's variable, like if you're talking about 10 samples, maybe it's not, but if you're talking about all public data, perhaps it will be quite common, like ability to analyze 70,000 samples or 100,000, million samples. So that's there. Inability to perform bioinformatics analysis, like training of students and mentees in terms of like what are the proper bioinformatics analysis and method development, things like that. Control access to certain data sets. I think that's hard to overcome. If you cannot reuse the data to the massive scale, that's a challenge. And incomplete metadata. I think I'm still surprised how it's not widely realized that, despite availability of the raw data, of beautiful raw data, if you don't share mortality, for example, or any other phenotype, but your goal of your project is to assess the mortality, the data sets become useless, right? You cannot train, you cannot do anything. So metadata is so important, but not yet to be widely recognized. So in terms of the barriers, like we were thinking about that. So I think educational barrier, like enabling students and like members of the community in terms of bioinformatics skills, I think that's possible, and we are on the mission. So we have thought about how to empower students with no computational background with computational skills. And I did it during my postdoc, talking about the Unix, and actually think about the globally. So like with Pavel Pevsner and others, we have published Nature Biotech paper about like what are the capabilities, like open data and bioinformatics and education in different parts of the world. And recently I'm curious to know, can we enable such culture at the school, like where I'm currently at the School of Pharmacy, with no kind of computational cultures whatsoever? Can we enable that, and what it takes? So we are on that journey. About the metadata, I think it's harder. I think, like there are a few possibilities what can be done, maybe mining the text of the publications, perhaps, but that's probably is condition that they do share the phenotypes of publication, but not all the time that's the case. So we have actually started that by publishing the editorial in genome biology, and we are continuing that. So that editorial, the numbers I have shared with you, that 40% of the metadata is not shared, was purely based on the sepsis RNA-Seq or beta-site transcriptomic studies. However, now we expand that across other diseases, and we have looked at the variety of the phenotypes. So this is the numbers you can see. So x-axis is the availability of the data in publication versus repository. So some phenotypes are good, like tissue type, but some are not that great, like mortality, clinical severity, ethnicity, surprisingly. People don't report that. And I think what you see on the box plot is quite nice in the sense that there are some publications which do share everything, but there is no single publication which share all the phenotype properly on the public repositories. And perhaps I don't need to emphasize for this audience that it's absolutely crucial to enable reusability. You can process 10 publications, 100 publications, perhaps, if you're diligent and kind of concentrated, but you cannot do that 1,000, not talking about tens of thousands, right? So you need the metadata on the public repositories to enable large meta-analysis or reusability of the data. I think there are other interests we have in the lab, kind of more like on the general side, kind of standardization of bioinformatics software. So previously we have talked about kind of what we call bioinformatics crisis, software crisis that many tools don't install. And we try to quantify that. So that receives quite a response from the audience in terms of the Twitter and things like that. I think I didn't check for a while, but we were like 21 by the Twitter, like preprint number 21 by preprint. So it was like basically quite large of the community reflected on those issues. I think bioinformatics education, we pursue that, and in particular kind of narrowing the education research gap and perhaps involving undergraduates in the research and training them and enabling or empowering them with research opportunities. I think that's on the list. We also talk about accessibility, diversity, inclusivity. And recently we have published the analysis about the virtual conferences, recognizing there is multiple challenges and limitations of those, and which I don't think the broad audience is aware of. So we have worked with different societies to ask, how was that having the having the virtual format, and how the revenue and things like that. But I think one analysis of the Recomp conference suggested that as soon as you do free of charge registration, that was the case for Recomp last year, basically looking at the map, like so many countries, especially developing one, individuals from those countries are able to join and get access, I guess, to the world science, so the science which is happening. So I think that's kind of breaks the barrier, geographical barriers, administrative barrier, so increase inclusivity. So that's something kind of generally also interested in. Okay, so just to be on time, so just to wrap it up, I guess I hope I was able to convince you that open data and bioinformatics method is the powerful tool to perform immunological research. I guess that's yet to happen, it's just starting. And I think what I focused today was assemble of human immunome and enable population immunogenomics through repurposing of other technologies, perhaps not kind of initially created for immunological research. And I guess like moving forward to comprehensive population-specific allele immunogenomic reference database across diverse populations. I think in terms of the future work, we're always kind of looking in front. So I think robustness and reproducibility of immunogenomics tools, that's something interesting for us. Like if you take the same sample and sequence that in Colorado, in California, like will you get the same results? And if not, whom to blame? Is it the technology, which is producing different results? Or is it bioinformatics, which is not able to handle certain alterations of the technology? Kind of doing 1,000 genome project for immunogenomics, that's, I guess, my dream or my ultimate project. Like can we recruit the population across the globe and learn about the distribution of like TCR and BCR alleles across the globes? And my understanding, that's the signal might be more stronger than the human genetics, because the immune system reflects the exposure of those populations to different pandemics and diseases and other things. And I guess like one kind of goal is to develop the methods which can infer the ancestry from AR-Seq, because as we can see, we cannot rely on self-reported ancestry because it's not shared, but also it's not that reliable. As we all know, people can be a mix, but not being aware of that. So genetic ancestry, this is ultimately the best way to measure the ancestry. And we would like to see if we can do that directly from AR-Seq data. Okay, just to wrap it up. So that's work of many people in my lab, postdoc, PhD students, and master students, and undergraduates, so work of many. And yeah, so thank you very much, and we'll be happy to answer any questions. Thank you.
Really great talk.
Thank you.
So I think we have time for questions. We do have one question in the chat, and then others can follow up. So Tanya asked a question about, in order to get a protein sequence for each read, do you translate all 3 open reading frames and then find the proper cysteine?
Yeah, exactly. We do convert to all the frames. And actually, we have checked that it's always, like there is no kind of possibility that you will have 2 kind of matches. It will be always 1 frame which will produce the correct result. So yeah, indeed, we check all the frames.
Cool.
I had 2 questions, one related to TCR sequencing and BCR sequencing. So the first about the TCR-Seq, as far as the data that you're acquiring, are you getting matched HLA types for those samples? And how useful is TCR sequencing without that matched HLA sequences?
Yeah, no, we actually don't look at HLA yet. And I think it's actually challenging to do it from from TCR-Seq data. At least I'm not aware of any methods able to do that. And actually, that's on the list. Probably I forgot to put on the future work. We would like to design ones which can infer HLA type from TCR-Seq data.
Yeah,
but
and then sorry, my second question about the BCR sequencing and trying to pull out new alleles based on ancestry, which I think is a really important thing to be doing, because I know that there's a lot of gaps in that right now. But I'm just curious how your group is going to be able to discern between allelic differences and somatic hypermutation.
Right, I think it's like, yeah, that's like actually one of the challenges, like, and I think I mean, perhaps, like maybe starting with TCR-Seq, right, where you would expect less somatic hypermutations, like that will be, we'll take the easy task first, because I imagine that will be quite challenging for BCR. But perhaps, yeah, recruiting like, I guess like maybe, I think it's like, I think, yeah, I think designing, kind of doing that bioinformatically. And I think one idea we have got, I think even like maybe generating different body sites or like different time points, like, and then kind of training the bioinformatics algorithms to differentiate between like, germline versus like somatic in that sense. But I hope that's possible. Yeah, that's one challenge, but I hope that's possible. And then, yeah, we're actively thinking about that. But again, think about TCR-Seq. I hope we will not have that extensive number of those as we will have in BCR-Seq, in BCR.
Thanks.
Great, thank you.
I have a question for you, Serghei.
Sure.
I'm curious about the educational piece where you talked about the need for big data computational skills more broadly. And I was curious what you thought the role of cloud computing in that was, right? So do we need to get compute as well as data more broadly distributed?
Yeah, thanks, Larry. That's a great question. So no, indeed, I think the cloud, if it's accessible, or it's kind of like people can afford that, right? Because when you think about like large scale analysis, I think we did calculations. If you want to process the whole SRA RNA-Seq, it will cost you, let's say, 50 cents per sample, but that's come to the 40K budget. Not necessarily very attractive, even for the people in US, I would say, right? So I think making that available, and I think Amazon is doing some grants. And actually, on the education note, I mean, I'm teaching some bioinformatics for the country. I'm originally from Moldova, which is like far away in Eastern Europe. And we got some arrangement for the medical school there. So we designed the course for them like online. And actually I was surprised to know some people know about the cloud and they're applying for the credit, and they got some, like credit from Amazon or Google. So they have resources. So, but I think I agree with you, that will be essential to kind of enable that. And I think it's unclear what will be the models, who should be paying for that. I guess, is it Amazon providing those resources? Hopefully they will continue to do that, like a small grants. But that's, like having the cloud, I guess that's all, coming to the discussion about like developing countries. I guess knowledge and access to the resources, this is all needed to enable bioinformatics research across the globe in that regards, like as soon as there is a knowledge and people are able to ask correct questions and they have access to the cloud, they can do cool stuff and reanalyze and run secondary analysis. I don't see any kind of other barriers, but perhaps, like cloud is the barrier which needs to kind of overcome it.
Sorry, I'm going to come in a little fuzzy here. But to follow up on that, do you think some of like the health disparity in terms of informatic knowledge across like ethnic groups or certain populations is partly due to the cost prohibitive nature of this compute resource in general to some of these more underrepresented groups in developing countries?
So just to, I got some hard time hearing that, but are you asking about like disparities in terms of access to the knowledge or like the bioinformatics skills? Was this question?
Yeah, so kind of like we see, for example, like European ancestries tend to also have the best reference sequences out there and stuff. Do you think part of that is due to the disparity in just general wealth of countries and ethnic groups?
Yeah, that's a great question. I think so. Like I think the countries which are generating the data, they generate the data which is from the major groups or from the groups which are dominant the countries, right? If it happened to be that many European countries are prosper, so they generated the data from themselves, or like from the people from those countries, right? Living, I mean, there is some initiatives in Africa and other parts of the world, but they yet to kind of get the full scale. So that's, I think, the big problem. And actually, that's an interesting question, like how the genomic databases, right, are representative of of those kind of minorities or underrepresented groups per country. We know globally is not great, right? We know globally that Europeans are dominating, but like per country, let's say if you take US and you take the whole genome data in the US, will be the fraction of Asians or the fraction of, let's say, Latino and other groups representative kind of from the number of samples, genomic samples, will that match the fraction of those individuals in as the fraction of the populations? And checking that per country, for example, that's interesting to me. But I think centralized until, like many countries are developing and lacking the resources to launch sequencing initiatives, I guess like we need kind of centralized efforts like we have in Africa, but because we need to scale across other groups.
Cool. I think we are perfect in time. Not sure. Casey, do you have final comments or questions?
It's okay. I'll ask him one-on-one.
Yeah, we have other meetings. All right. Thank you. Very nice talk. Thank you, everyone.
Okay. Thanks a lot.